form 3 molecules of ATP. So at the end, in cytochrome oxidase last enzyme, you've got 4 electrons plus 4 protons which come forming ATP, and then there is molecule of oxygen to end up with water, and also they form ATP, and well, around about 50% of the energy is liberated as heat. So this is oxidative phosphorylation taking place. So I told you the reactive oxygen species are related to oxygen, but they are reactive. Why are they reactive? Again, I have told you they are reactive because they have got a single unpaired electron. If there were 2 electrons in opposite spin, it would have been non-reactive, but when there is only 1, it would try to complete the other one, so it would attack, and to attack the most vulnerable things. Well, the good effect is that they kill these microorganisms. This is good, but when they are in excess, for example in oxidative stress, then they would attack the normal body tissues. So they have to be kept in control. How they are kept in control? By superoxide is kept in control by superoxide dismutase. This H2O2 is converted to OH and also kept in control by glutathione in the RBCs that I am just going to talk about. This OH is also controlled by glutathione and converted to this. So this was in detail about these enzymes. And hydrogen peroxide itself is kept in control, kept in check by catalase. Superoxide dismutase is again of it contains a cofactor. The cofactor is copper in the cytoplasm on the cell, extracellular. Then it has got manganese in the mitochondria, where it also has a cofactor of zinc in the cytoplasm. And so in the cytoplasm, it is copper and zinc. Extracellularly, it is copper. These are the cofactors for superoxide dismutase. H2O2, let's focus on H2O2. H2O2 is converted into hypochlorous acid. H2O2 also forms hydroxyl ion, and H2O2 is also acted upon by catalase and converted to water. Because in catalase, 2 molecules of H2O2 will give rise to 2 molecules of water plus oxygen. So this is how it is kept in check. Oxygen. So when it is converted into neutral water and oxygen, so no chance of making it this potentially lethal. This is the most lethal of that. Lethal hydroxyl ion. And also H2O2 in RBCs. H2O2 is converted to, is checked upon by glutathione. Reduced glutathione, which has taken this hydrogen from NADPH to form HMP shunt, and it is converted into 2 molecules of water. So this is how the RBCs are saved. But if there is G6PD deficiency, because this, then there is no NADPH, NADPH2. When there is no NADPH2, no glutathione reductase will be formed, H2O2 will be in excess and it will cause the breakdown of RBCs leading to hemolytic anemia. So, I would like to amplify a couple of things. Now, what happens in RBCs? I will just explain it a bit. So you've got all these enzymes, the reactive oxygen species I've told you, and the antioxidants. Antioxidants. Now what are the antioxidants? The enzymes I've told you, they are superoxide, dismutase, then you've got catalase, then you've got glutathione peroxidase, superoxide dismutase, catalase, glutathione peroxidase. Then you have got the vitamins, vitamin A, that is beta carotene, then you have got vitamin C, then you have got vitamin E. So these are the natural antioxidants, and some artificial antioxidant that you add in food, antioxidants added, artificial. Hydroxy, butyl, toluene, so these are artificial added in food. Superoxide dismutase acts on these superoxides and converts it into 2 molecules of water. So finishes it. Catalase acts upon 2 molecules of H2O2 and converts it into 2 molecules of water plus oxygen. So the free radicals are gone. Glutathione peroxidase. There is, I just do it in detail, I have given it previously. This is present in, rich in RBCs. It's present in red blood cells, RBCs. What does it do in RBCs? In RBCs, there is HMP shunt. It helps in the production of NADPH2. This H2 goes to glutathione, which is present in the cells, and it forms glutathione reduced. So 2 molecules of glutathione plus H2O2 will be converted into glutathione plus 2 molecules of water. So RBC, normal RBC. If there is defective HMP shunt or there is decrease in G6PD, glucose 6-phosphate dehydrogenase, then NADPH will not be formed. Then no NADPH2, this will not be formed, and so glutathione will not be reduced. Glutathione not reduced, leading to H2O2 in excess. This will cause hemolysis of the RBCs. So there will be hemolytic anemia. In G6PD deficiency, there will be hemolytic anemia. Action of vitamin C, E and vitamin A. I will sum it up. These antioxidants vitamin E, tocopherols, they are present where there is high increased PO2 levels. Increased where there are high PO2 levels. High PO2 levels are present in lungs, and the RBCs. So their membranes, vitamin E tocopherol is present in the membranes lung RBC at high partial pressure, while vitamin K and vitamin C are present where there are low PO2 levels. Now, if there is a reactive PUFA radical which has been acted upon by a reactive oxygen species, what will happen? That tocopherol will come to the help. By providing its H. This H will go and provided with an electron, so that single electron would be covered up, and so the chain reaction stops, terminated by giving it H, that is one electron. So the orbit is outer orbit is complete. Now then it would get oxidized. So to keep it functioning, it must be supplied with H from vitamin C. Vitamin C, that is ascorbate, and vitamin C gets its H from glutathione. H. reduced glutathione. And glutathione gets its H from NADP. H2. And this one gets it from HMP shunt, hexose monophosphate shunt, which is present in all cells. So all the cells, they've got an HMP shunt, and the important enzyme is G6PD, glucose 6 phosphate dehydrogenase. This gives rise to NADP H2. This one reduces glutathione. This one reduces vitamin C. Vitamin C gives its hydrogen to tocopherol. This one gives hydrogen and stops the chain reaction in PUFA. So this is all the chain for antioxidant reaction. This is all antioxidant protective reaction. To avoid any confusion, these PUFAs have been acted upon by one of the reactive oxygen species, which took its electron, leaving it one solitary electron, unpaired electron. So itself it has become a radical, and it is attacking other chains and damaging them, and so the chain reaction is going on. Cell membranes are broken down, cell disease, then organ disease, then the person gets diseased. So this is the science behind, this is the pathogenesis behind aging, disease, aging, mutation, etc., etc. So this electron must be, one electron must be provided. So this is how the body tries to repair the damage to PUFA by reactive oxygen species. I think I made it clear. So you've got a whole series of action. HMP shunt, that involves glutathione and vitamin C. Vitamin C replenishes toco, and toco action supports it. So this is how this antioxidant is taking place. Because the reactive oxygen species are very dangerous. I've told you they are acting upon PUFAs of the lipid. This is called the lipid peroxidation. So antioxide, this is lipid peroxidation, has been stopped by this chain reaction. How they are produced, I told you in the previous drawing on the board. And all these are being produced in the body all over, and the body is trying to control them. Well, reactive oxygen species, this is called lipid. Well, they can also affect the DNA. They can also affect the DNA. Especially they attack guanine. And they cause the change, they cause the addition of hydroxyl group to guanine. They've got the addition of hydroxyl group to guanine at position 8, and this can lead to mutation. Guanine. Reactive oxygen species, guanine. So if I draw guanine over here, over here there is the addition of hydroxyl group. So, and this can lead to mutation in guanine. At this carbon there is the addition of this OH. So also reactive oxygen can affect certain proteins and DNA, etc. But I'm told you about the lipid peroxidation. The lipid peroxidation surprisingly affects what? PUFAs. And I've told you PUFAs are present at carbon number 2 of the phospholipids. Where are they present? They are those polyunsaturated fatty acids which are located at carbon number 2 of the phospholipids. Which phospholipids? Of the cell membrane, because cell membrane is made up of phospholipids predominantly. So if they are damaged, the phospholipid is damaged and the cell membrane is damaged. So I've repeated it over and over again. So this is how the reactive oxygen species are produced. They have got unpaired electrons and they are causing so much damage. They are of benefit as well when they are related to the phagocytic activity in the polymorph nuclear leukocytes and macrophages. Because the formation of superoxide is called respiratory burst, and NADPH oxidase is the enzyme. Respiratory burst kills the microbes. Then hydrogen peroxide formed in the cell is also converted to hypochlorous acid, again with the help of superoxide. Hypochlorous acid is another compound called as bleach, and it also is activated, is formed by, as I showed you previously, by the enzyme myeloperoxidase. It also helps in killing the bacteria. So the microorganisms are killed again. So all these are involved in antibacterial activity. But unbridled, excessive production of reactive oxygen species will cause damage. For example, also in homocystinureas, when there is a deficiency of transsulfuration reactions are not taking place. There is deficiency of vitamin B6, and also there is deficiency of cystathionine beta synthase. There is hyperhomocystinuria. The increase in homocysteine levels will also increase ROS, reactive oxygen species, and in that case they affect the endothelial cells because LDL cholesterol is oxidized, LDL oxidized, again it will affect the endothelial cells, cell membrane, it will be roughened, and again there will be process of atherosclerosis. So it is the reactive oxygen species which is damaging the tissues whenever in excess. So the nature is kept in check because of different enzymes present in the cell as well as in the extracellular tissues. And also liberal intake of vitamin A in the form of beta carotene, vitamin C and vitamin E will do the job. So the end result is a balanced diet, healthy balanced diet with lots of